Hello everyone, and today's topic are the central dogma of life. Means it is discovered by the Francis Crick in 1957. It is a process by which instruction in DNA are converted into functional product. In simple word, that DNA forms a protein. Within each cell of the genetic information flows from DNA to RNA to protein. This flow of information is unidirectional and irreversible. The information carried within the DNA dictates the end product, that is protein, that will be synthesized. This information is the genetic code. Conversion of DNA encoded informations to RNA is called as a transcription. The information from mRNA is translated to the amino acid sequence in the corresponding proteins. What is meant by transcription? The process by which the genetic information is converted from a double stranded DNA molecule to a single stranded RNA molecule. Only one strand of DNA serves as a template. This is the transcripted or antisense strand. The complementary strand has a sequence additional identical to the RNA sequence, except for uracils in place of thymine. It is called as sense strand or RNA like strand. Next is a DNA transcription. Transcription is the first step in a gene expression. It involves the copying gene's DNA sequence to make a RNA molecule. Transcription is preferred by the enzyme called as RNA polymerase, which link nucleotides to form a RNA strand using DNA strand as a template. Transcription has three steps. In that, first is the initiation, second is the elongation, and third is the termination. In eukaryotes, RNA molecule must be processed after transcription. They are spice and have a 5 cup and polytail put on their end. Transcription is controlled separately for each genes in your genome. Stages of transcription. In that, first is the initiation. RNA polymerase bind to sequence of DNA called as a promoter found near the beginning of the gene. Each gene has its own promote only bound. RNA polymerase separates to DNA strand, providing the single stranded templates needed for the transcription. These all about the initiations. Next is the elongations. On strand of DNA, the template strand act as a template for the RNA polymerase. RNA polymerase, that enzymes which link the nucleotides to form a RNA strand. As it reads this template on one base at the time, the polymerase build up RNA molecules out of complementary nucleotide, making a chain. That groups from 5' to 3'. The RNA transcript carries the same information as the non-template strand of DNA, but it contains the base uracil instead of thymine, that is T. This is all about the elongation. Third step is the termination. A sequence called terminator signals that the RNA transcript is complete. Once they are transcribable, they cause the transcript to the release from the RNA polymerase. DNA and RNA and their types. DNA, in that it is a deoxyribo acid. It is an organic chemical consisting of genetic introduction and information for protein synthesis. DNA can be seen in a mast cell of all the living creatures. It is a vital part of the reproduction which genetic hereditary occur. Therefore, the objective is to transfer the inheritance from the order generation to future generation. Nuclear DNA is nothing but the DNA present within each cell of nucleus. DNA is a group of uh, molecules that is responsible for carrying and transmitting the hereditary materials or genetic information from the parents to us offspring. So this is all about the translations and transcription. Next is the difference. What are the difference between the replications of DNA and transcription? 1 minute. In that replication, in it is the process of formation of two copies of DNA molecule. Entire genetic DNA is copied. Transcription is the only segment of DNA is copied to RNA. It is the synthesis or formation of DNA from RNA. Transcription is synthesis of RNA from DNA. Both the strands of DNA takes place in replications. Only one strand of DNA acts as a template strand and get transcript to RNA. Nitrogen bases found in DNA polynucleotide chain are adenine, thymine, guanine, and cytosine. In transcription, the polynucleotide chains are adenine, uracil, guanine, and cytosine. Replication, it is catalyzed by DNA dependent DNA polymerase enzyme. In transcription, it is catalyzed by DNA dependent and RNA polymerase enzymes. So this is all about the transcription.